Some of the best scientists in the world have observed this image, measured, studied it, and still have no explanation for how the image was formed.
The most studied artifact in the world is the Shroud of Turin, a relic steeped in mystery and controversy. I am very skeptical about this. It is believed by many to be the burial cloth of Jesus Christ. Dr. Kelly Kearse, with a background in cell biology and immunology, became deeply intrigued by the shroud's bloodstains.
I believe new evidence shows that we can now really not definitively say that it's human blood that's been demonstrated to be on the shroud. It certainly could be, but I think that those things have to be taken into consideration based on what we now know in the 40 years since those original studies were done.
His research posed questions that challenge majority belief. Is the red substance truly blood? If so, is it human, animal, male? These questions led Dr. Kearse down a path of scientific inquiry, where he uncovered surprising findings that challenged established theories.
And as well, that the high bilirubin levels were not responsible for the reddish color being maintained with time. So that is still something that remains very interesting and very mysterious.
Beyond his work on the shroud, Dr. Kearse has an impressive academic background with postdoctoral fellowships at renowned institutions such as Johns Hopkins University School of Medicine and the National Cancer Institute. In 2015, he saw the shroud in person with members from the STURP team. He also met with Dr. Alan Whanger and got to view sample threads of the shroud under a microscope, where he looked at the slides of the bloodstains. Today, we have the privilege of talking with Dr. Kelly Kearse to discuss his groundbreaking work and shed light on the mysteries of the Shroud of Turin. If you could tell us your backstory of how you became interested in the shroud.
In the shroud. Okay, so first time I ever heard of the shroud, I was in college. I was working at a grocery store, was on a break, and I saw a magazine. I'm pretty sure it was National Geographic. It was in the early 80s and had quite a few photographs of it. And the one of the negative shot of the face kind of grabbed my attention. And I looked through it. I thought it was interesting. I honestly didn't think that much more about it. Through the years, I watched a couple of television programs. I think there was Unsolved Mysteries. I watched that. I read a few paperback books, but kind of a casual interest in it. Then in the year 2000, I was going to Italy for a scientific conference. And I thought maybe on a day off, I might be able to take a trip over to Turin. I knew it wasn't on public exhibition, but I was kind of intrigued by it. The conference was pretty jam-packed, so I never ended up taking that trip. And then in 2010, the shroud was going on public exhibition, and the dates looked really close to when my spring break was. So I thought, well, maybe I can go see it. And as the kind of more and more was announced, it was just a little bit off. So I wasn't able to make that trip. But in preparation for it, I began to read articles about it and especially listen to a lot of podcasts. And my former research background is in cell biology and immunology. So the studies about the blood are kind of what really stood out to me. And I heard some things that didn't kind of scientifically didn't really make that much sense. So I sort of investigated it. And the more I investigated it, the more questions seemed to come up. And then, so I really kind of got into it for a full steam back in around 2010, just in preparation for that trip. Now, I ended up not being able to go on that trip, but in 2015, there was another exhibition. I was able to go on that one. What was kind of cool about that one, I ended up going with a group that was led by Dr. John Jackson, who headed the STURP team. So I got to spend, we traveled throughout Italy, but got to spend the whole week with him, and we would talk together about the shroud. And then when we went to the exhibition, you can go in the main part and see the shroud up close, and then you can go to the back of the cathedral and stay as long as you like. So I actually had several ticket times to go through and look at it up close. And then on some of the ways back, I actually got to go over there where it was just me and Dr. Jackson, and that was really cool. So that's when I got to see it in person in 2015. And then I was fortunate enough through Dr. Alan Whanger, who had some shroud samples, microscope samples that he'd gotten from Dr. Max Fry. I was fortunate to be able to look at some of the threads from the shroud under a microscope, to spend an afternoon down in his basement looking at the particularly slides of the bloodstains.
What's up, everybody? So when this interview was over with Dr. Kelly Kearse, I was very frustrated at myself because I did not ask some questions that I wanted to ask. I don't know, I just forgot to ask them. So I contacted Mr. Kearse and asked him if he could answer about 9 extra questions. And this is about the shroud samples that he examined. And so what you're about to watch is those answers to my 9 questions, and then we'll get back to the original interview.
Okay, I've been asked to provide some answers to some supplementary questions, particularly about looking at the samples with Dr. Alan Whanger. So question 1 is, can you provide specifics about the samples you examined, including which body parts they were associated with? Yeah, the samples I was able to look at, I looked specifically at the samples from the forearm and from the back of the foot. Number 2, approximately how many samples did Alan possess from the Max Fry collection? I believe there were 27 slides total in the collection. Number 3, were you one of the select few individuals granted the opportunity to examine these samples? I guess so. I believe that Dr. Alan Adler, Dr. Whanger, of course, Paul Maloney, of Assist, they examined them, but I don't think a lot of people did. But yes, I was fortunate enough to be granted the opportunity of it. Number 4, were any definitive conclusions drawn regarding the presence of blood upon examination? So all I did was just look under the microscope. The one thing I remember is they looked more reddish than I expected. That was the main thing that I remember from that. It's been some 12, maybe 13 years or so ago, but that's the thing that sticks with me. Number 5, with the passing of Alan Whanger in 2017, what is known about the current whereabouts of these samples? I myself do not know their current whereabouts. Number 6, what prompted Max Fry to entrust these samples to Alan Whanger? My understanding there is that Alan Whanger and Paul Maloney of Assist, they in fact purchased these samples from Max Fry's widow. That's my understanding of that. Number 7, did you personally request permission from Alan Whanger to examine the samples? I sure did. I wrote him an email, and he was very kind to respond to me, and responded very quickly and invited me to his home, where I met Dr. and Mrs. Whanger. Both were extremely nice and very, very cordial, very welcoming, and went down into Dr. Whanger's basement and examined the samples, and we talked about quite a few other things related to the shroud. Number 8, were any formal tests conducted on the samples during your examination? That's a no. And number 9, is microscopic analysis the sole method by which these samples can be observed? Can they be seen with the naked eye? You can look at the threads with the naked eye, but you're not really going to see much. Under the microscope is really what's going to give you any kind of information there. So microscopic analysis, as far as observation, that would be the best route for that.
And you were able to take a picture of the shroud when you saw it in person?
Yes, I was. So they kind of, as long as you were pretty discreet and pretty reverent, they were okay with it. It kind of depended on who was sort of running the line at the time. I had a really small, discreet point and shoot when I was up close that I took some pictures of it. And then at the back, I had a camera with a little bit bigger lens, and they would let you take a few pictures. Then they would, kind of say, okay, that kind of thing. And there were like several times I went through and didn't take any pictures at all. So it kind of depended on who was in charge of the security at the time. So kind of discreetly, you were able to. I was pretty surprised how close we were able to get to it when we went, about 12, 15 feet, give or take. And the first time we were able to stand there at least about 5 minutes. So got some really good looks at it. And I would kind of go through, I would go through one time and I would say, okay, I'm gonna really concentrate on the bloodstains here, the side wound. I'm really going to look this over, et cetera, et cetera. And then maybe the second time it was, like no science. I'm just going to go in there with a faith on this and just kind of meditate on this. And, so I sort of did a little bit of both as I went through.
And I guess there was a rumor that in 2025 they would also have another event where you could go see it, but I guess they canceled that one.
I heard that might be a possibility, but the latest I've heard is this is all unofficial rumors that the public exhibition, I don't think that's going to happen. But I have no connections or no idea, just a rumor.
Is real blood present on the shroud? Do we know that there's actually blood on it?
So I would say that's scientifically, my opinion, that's a definitive yes. I would say there have definitely been blood components that have found to be there. Hemoglobin through a variety of tests, both chemical tests, also spectroscopy, various light type tests. So I think that's been shown pretty solid. Then also kind of the strongest evidence for me, having an immunology background, being an immunologist, is there's immunological evidence for blood components such as albumin and immunoglobulin. So I would say, yes, those are definitely there for sure.
What tests did the STURP team do? Do you think they were good tests on the blood?
Absolutely. Yeah, absolutely. So the STURP team, this is back in the late 70s. And I think the tests that they did use the best tests that were available at the time. And they're also up against really a lot of limited materials. So I think that's something that's really, really important to keep in mind. And the tests, the most of their focus as far as the blood goes is to see, well, are blood components really there? So I think they did that quite thoroughly. Hemoglobin, bilirubin, immunoglobulin, albumin. And then you start to get in, and that stuff was published in peer-reviewed journals. I think that works very solid. And then you kind of start to get into the nitty-gritty of, well, blood's there, what type of blood is it, et cetera. And you have to bear in mind that all that work as far as what type of blood it is, none of that has ever been published in a peer-reviewed journal. It's more of a work in progress type thing. And again, they're up against limited amount of sample. And also, hindsight, 40 years later, hindsight is 2020. So yes, I absolutely believe they did very good tests. A lot of respect for their work. You bet.
Why did they come to the conclusion that it was human blood or male blood, for that matter?
Okay, so let's we'll do the human part first. So you have two groups that are going to test for the type of blood. You have the STURP team, so Dr. Heller and Dr. Adler, and then you have an Italian scientist, Dr. Baima Poloni, who also took some samples at the same time. So we'll start with Heller and Adler first. So they went for albumin, which is the major protein in blood, and they had an antibody that was made in rabbits against human albumin. So you purify human albumin, you inject it into a rabbit, and that rabbit produces antibodies. So then you can't see the antibodies with the naked eye, so you label it with a fluorescent tag. And then if you look under fluorescent microscope, wherever that lights up, you know that it's found its target, which would be albumin in those bloodstains. So STURP does fibers without blood, which are negative. They do fibers with blood, which are positive. It lights up. This suggests that you have albumin present in those bloodstain fibers. Now, what type of albumin is it? Well, the rabbit's immune system responded to human albumin. That's the protein which the antibody was generated against. But it doesn't mean that that's the only type of albumin that the protein is going to recognize. So if you just kind of flip that slide back up that you had a second ago of the rabbit there, bingo. So if you look at the top, the top. So in separate experiments, they tested that antibody against human albumin, which it was positive, as you would expect. It also reacted with blood from chimp, which is not really unexpected. And you saw some reactivity, it's not shown in this slide, but some reactivity with baboon. They also tested for cow, which is shown here, and also tested for horse, which were negative. So the conclusion from Heller and Adler originally was that it's at least primate blood. And again, this was never published in a peer-reviewed journal. It was kind of more of a work in progress. So we have at least primate blood from their studies. Well, around a similar time, Bologna tested for the presence of another blood protein, immunoglobulin or antibody. He used an anti-human immunoglobulin as the inoculate into the animal. He saw positive reactivity, and he said, well, this shows that it's human blood, because on the vial, it'll say, let's say, rabbit anti-human. Now, when you see that on the vial, that's what it's generated against. It does not mean that's the only type of blood that it reacts with. That's called cross-reactivity when it reacts across species. Bologna never addressed that. I would be, as an immunologist, I would be way more concerned about the cross-reactivity with the anti-immunoglobulin, which he did in his studies, because it's known that those proteins are very highly conserved among species. So I don't think you can claim at all from his work that it's human. Cross-reactivity is never addressed. Now, Heller and Adler did address it in their studies. But what we now know, if we just go back to that slide one more time, what we now know some 40 years later is that this antibody that they use would have reacted with species that were originally not tested, such as those shown at the bottom. So these antibodies would also react with blood from rat, from mouse, from cat, from dog, and several others. And if you look genetically at the genes for albumin, which have now been sequenced through various genome projects across albumin from different animals, it all makes sense. Cat albumin is around 80% or so similar to human. And some therapies in dogs, you can actually put human albumin in dogs to help correct them if they have low albumin in the blood. So it's very similar among those species, quite a few of which weren't tested. So I think if you put all this together with hindsight and looking at what was originally done, so what was originally done, it was, well, it's at least primate blood from the STURP side, and then Bologna, it was, well, it's human blood. That's what's on the label. You have to take cross-reactivity into account, which Heller and Adler did. The primate blood sort of just morphed over the years into just human blood. One person will say it, then the other just kind of repeats what they've heard, then the other repeats, and so forth. So it just kind of eventually became human blood. But I think if you look at all the data from a scientific standpoint, I think it takes it back to square one. I think scientifically, the type of blood, what species it's from, it's currently unknown.
So you know for sure it's not cow and horse?
That would, yes. So looks like that's very good, yes.
Okay. Is there any other animals or anything else?
That you could rule out? They might have also tested pig, if I remember correctly, but I would have to go back and look that up. And I don't, let me just say, I don't necessarily believe that it's rat blood or something like that that's on this route. I think if someone were going to add blood, it would be very easy to get human blood to do that. I'm just talking about from a scientific standpoint. If you want to say that it's human blood, which you'll see it a lot on the internet, you'll hear it in a lot of YouTube talks, the blood has been shown to be human. I think if you're going to say that from a scientific standpoint, the data has to be there to back it up, which in my opinion, it's just not there. It's unknown.
They concluded that the blood type was AB.
Right.
And how did they come up with that?
Okay, so this is Bologna's work, and so he's the one that did the blood typing of the shroud from the samples taken around the same time. So the blood groups were discovered in 1901 by Dr. Carl Landsteiner, who was an immunologist. You have four general blood groups: type A, type B, type AB, and type O. Most of the people in the population are going to be type O, around 47, give or take percent. And the percentages vary depending on the geographical region. In a close second place is type A. And then you get down to around 8% are type B. And 3 to 5% are type AB, although again, it's going to vary depending upon geographical region. So the way you test for blood type, there are two ways you can do it. Well, there's two ways you can do it with antibodies, which was what was done for the Shroud. There's, let's just say you can test it serological using antibodies, or you can test it on a DNA level. And the DNA level tests were not done for the Shroud. So we'll just stick to the antibody testing for blood type. So antibody testing, you can test that in two different ways. There's something called forward typing, where you test for the presence or absence of those AB molecules on the surfaces of red blood cells. There's also reverse typing, where you can test for the presence or absence of antibodies in the serum. So we'll take the forward typing first, because that's kind of the most common that's done. It's very similar to what we described for the albumin test. You have antibodies against the A molecules, the B molecules. O type, O means without. So it doesn't express A, it doesn't express B. AB would express both A and B. A, of course, expresses A, B expresses B. So Bima-Bologna tested using forward typing, labeled antibodies tested for those molecules on the bloodstained fibers and reported that they were AB. As soon as the results came out, shortly thereafter, Alan Adler of the STURP team objected and said, well, what do these results really mean? Because we know that a lot of older material types as AB. It's not an unexpected result to have AB come out to be the blood type. Now, why would he say something like that? The reason, what it's based on, is that these AB blood antigens, they're not unique to human red blood cells. They're carbohydrates or sugars. They're found all across the animal kingdom, especially in bacteria and fungi. So if you have just even a little bit of contamination, which is typically an issue with older samples, you're going to get a result of AB. You could have a positive test for AB without a red blood cell being within a mile of the building. So Adler is the first person to really wave the flag and say, wait a minute, how sure are we really of these typing results? He didn't really buy it, because a lot of old material types as AB when you use these types of tests. The other type of test that Bima-Bologna did is called reverse typing. This one's a little more complicated, but here you look for the presence or absence of antibodies in the serum. So if you're a type A, in your serum, you'll have antibodies against type B. If you're type B, you're going to have antibodies against type A. If you're type O, you don't have A or B on your blood cells, you'll have antibodies against both A and both B. If you're type AB, you won't have any anti-A or anti-B antibodies. So the immune system is always going to create antibodies against the molecule which the person themselves doesn't have. So we do reverse typing, look for the serum, and this is unlike the forward typing. This is a functional test. Those antibodies have to be functional in order to work. And to ask that of an antibody that's several hundred or several thousand years old, that's very problematic. Reverse typing typically isn't even done with older samples, but Bima-Bologna did those type of tests. And when the result is AB, you don't really know if it's truly AB and those antibodies are in fact absent, or if with time they were originally present and then their function has begun to wane such that it's non-functional. So when the result is AB, you cannot tell if it's truly AB or is just an older sample to give you a negative result. It's very, very common to get that. It's kind of a catch-22 when it comes to the AB type. It's unknown by reverse typing what the results mean. So if you put both of those together, there's a tremendous amount of doubt of whether the sample is truly AB or not. And again, typing studies, they were never published in a peer-reviewed journal. I'm sure in an immunological journal that would have been pointed out. So I think you have to take those results with a very, very large, hefty grain of salt. And a lot of the relics that are believed to be been in contact with Jesus, Eucharistic miracles, these types of things, and I'm just talking about the blood typing of the Eucharistic miracles. Those results across the board, they usually come up as AV is a reported result. Not surprising that that's going to be the results that they find because of these false positive kind of issues. And you have to look at where this kind of stuff comes out. It's usually reported, but a lot of times no data is shown. Essential controls are absent. It's not in the how you would typically expect scientific results to be presented in a journal and peer review. And then you also ask about, I think, the blood being male. How do we know that the blood is from a male? That's from work by Dr. Garza Valdez. Again, never published in an article. It was published in a book that has a great title. It's called The DNA of God. So it's a really catchy title, but unfortunately the science is not very sound. So what was done there? He took some bloodstain fibers and he tested for the portions of 3 genes. One was a beta globin gene. This is a subunit of hemoglobin, which you find in red blood cells, the protein, and also presence of the X and the Y chromosome. So let me just kind of mention something that you often hear with that. So you often hear, well, he tested for the presence of those 3 genes, but the rest of the DNA was too degraded to do anything with. So when you hear that kind of thing, scientifically, it's a big flag that the person that's reporting this really doesn't understand exactly how the technique was done. So what Garza Valdez did, he used a technique called polymerase chain reaction, or PCR. And what you do in that technique, you make a probe for whatever particular gene that you're interested in. That probe is going to find that gene. It's going to totally ignore everything else in there. So they specifically targeted these 3 genes. The rest of it wasn't ever looked at. And, if you think about it, how is it that these 3 particular genes that they're interested in, a beta globin gene and the X and Y, why would large amounts of those survive and everything else would be degraded? It doesn't really make a lot of sense. And I think it's just a misunderstanding of how the technique was done. PCR technique is going to specifically target those 3 genes of interest. So let's take the first one, beta globin, a subunit of hemoglobin. Garza Valdez gets a positive signal for that. So he says this shows that the DNA signal is coming from blood cells. Incorrect. Skin cells also have a beta globin gene. They don't use that to make hemoglobin protein. It stays as DNA. It's never made into a protein in skin cells, but they still have the gene. So if you see a positive signal for that, you don't know if it's from a blood cell. You don't know if it's from a skin cell. You don't know. And if you go back and look at a lot of the photographs of the STURP team and others that were working on this route, this is late 70s. It's pre-DNA, before DNA science really took off. Nobody's wearing gloves. Nobody's wearing masks, hairnet, protective suits. So you're going to have some pretty widespread contamination there. It's known that hundreds and hundreds of people have handled the shroud throughout its history. There's no way to tell whether the DNA is truly coming from white blood cell or if it's coming from a skin cell. So when you have that positive signal, you can't say that it's coming from a blood cell, just like you can't say that the X and Y, the signal from the X and Y chromosome, is also coming from a blood cell. Simple contamination could very easily explain the results. In a single day, one person sheds around 100,000 or so skin cells, some of which contain DNA, which could be transferred, called touch DNA. So you can't really say that the blood is from male. Female DNA has also been found on the shroud as well, but there's no evidence at all that the DNA signal comes from blood cells. And let me just point one thing out. So often with DNA, it's kind of, it's talked about, well, there's a lot of contamination there. So it's written off that you couldn't do anything else with DNA. All DNA tests, there's really no purpose of them, etc. Now, I'm not a DNA expert. I'm not a blood expert. I guess if there's any expertise, I am an immunologist, but I'm not an expert in a lot of these things. I mean, I have had some experience at the lab bench with these, but you can have a situation if one thing you're going to be up against with something is old as the Shroud, hundreds of years old, thousands of years old, is DNA degradation. So you don't expect the DNA to be in fantastic shape, but today with modern technology, with the sensitivity of it, they're doing a lot with just a very, very small amount of material. And you don't always need a full-length gene to know that that particular gene segment is present. So you might be able just to have a very, very small piece of it. But just like if you hear a few notes of a song, sometimes that's enough to recognize it. Or you read just one line in an entire book, and you can tell exactly what book that particular line came from. You might be able to have just a very small stretch of DNA. And again, it all depends on how much is degraded, but even with a small amount, you might be able to do something with it. Now, it turns out in blood cells, particularly white blood cells, they rearrange their DNA as part of their normal maturation and development for their receptor genes. They're the only cells in the body that do this. Skin cells don't do it. No other cell does it. Skin cells will have the genes for these immune receptors, but they stay in an unrearranged form. In the lymphocytes, in the blood cells, the DNA is going to rearrange. It looks very, very different than it does in skin cells. So you could have hundreds of thousands of people that have touched the Shroud in that area. If you use a probe for the rearranged version of the gene, that probe will go in and find it. Again, if the DNA is degraded and there's enough to see, that probe will find it amidst all that contamination that you see, because the only rearranged version you're going to have is truly a signal from the blood cell. So that's something that could be done, even though scores and scores and scores of people have touched and contaminated. You could potentially sort that out if DNA was in enough a good shape to do that. And why would you do that? Well, it would show that the DNA signal truly does come from blood cells. It could also potentially identify the species of the blood, depending on the region that you targeted. It might be able to sort that out as well. So I just wanted to kind of include that, because often DNA is sort of pushed to the side. There's also mitochondrial DNA, which, unlike nuclear DNA, which is usually what is looked at most, which is only 2 copies per cell, mitochondrial DNA is about 100 to 10,000 copies per cell. So the copy number is much, much higher. So in cases where DNA is believed to undergo a lot of degradation, mitochondrial DNA could be an avenue. It's a totally different set of genes than the nuclear genome. It's about 37 genes. But that's another thing that you might be able to tell relatedness between a blood stain on one article and a blood stain on the other. So to kind of sum up a very, very long answer to the question about the male, I think there's no scientific evidence whatsoever. That is not scientifically valid.
So is the Shroud so contaminated where we wouldn't be able to get an accurate DNA analysis at all?
Well, so again, I think it depends on what you look for. The type of test I was talking about just a minute ago, the rearrangement, it wouldn't matter. If you're looking for things like blood type or something like that, contamination could be a very huge issue. But I think there are ways you might be able to get around it. So maybe you could sample from the reverse side, or better yet, I think you could go into the interior of it and take some samples from those interior threads. I think that's your best shot at trying to avoid contamination, if it were doable. Again, all depends on the extent of degradation. So if you're looking for, it depends on what you're looking for. For some tests, contamination, it's really not an issue if you're going for gene rearrangement that's unique only to those particular type of blood cells. If you're looking for kind of more general things, such as the X and Y and things like that, yeah, it could be, but maybe there's ways around it, or you could stack the deck in your favor a little bit more.
Why is the shroud reddish in color?
Okay, yeah, that's a great question. There's kind of two main schools of thought there. One is that the blood is red because of the blood, a property of the blood. The other would be, well, it's a property of the cloth, and that when you put blood on there, the cloth is treated as such that it's going to, the end result is the blood is going to stay red. So we'll take the something about the blood is unique. We'll take that one first. Kind of the primary one there is called the bilirubin theory from Dr. Alan Adler. And you'll sometimes hear in these discussions that bilirubin is hemolytic, that bilirubin lyses blood cells. That's not actually true. So that's a misunderstanding of how it works. So when red blood cells undergo a lot of turnover and are degraded, they will bust open, which is the hemolysis part. They will lyse open. They'll release hemoglobin. When hemoglobin breaks down, bilirubin is a byproduct of hemoglobin degradation. So it's not that the bilirubin causes the hemolysis or hemolysis. And I've asked actually several different bilirubin experts that's all they do is work with bilirubin about that. They said that that's just not true. I've done at the lab bench, I've added bilirubin to red blood cells to see if it causes lysis. It doesn't. So it's kind of just a misunderstanding. It's actually backwards. Bilirubin is generated from hemolysis. So when red blood cells break open, they release hemoglobin, which is broken down into bilirubin. So that's how the bilirubin is generated. So Adler believed the man on the shroud, because of undergoing such harsh physiological stress, he would have a lot of red blood cell turnover, a lot of hemoglobin release, breaks down a lot of bilirubin. And he would report that the bloodstains contain bilirubin, and he believed quite a bit of it. So he said that he could take bilirubin, add it to fresh blood, so not a separate experiment from the shroud, add it to fresh blood, and the blood would stay red over time. So he could repeat the kind of coloration that you have on the shroud. So I did similar experiments. I took bilirubin at various doses, including he had some really high-end values that he was using. I did all the way up to that and everything in between. There are two different forms of bilirubin. I use both forms, alone and in combination. I never saw that the blood stayed reddish. In my experiments, it always turned brown. I also looked at a particular type of rat called the gun rat, GUNN, that carries a mutation that the result of that is it has high levels of bilirubin in its bloodstream. So I looked at blood from a wild type rat, blood from the gun, and blood did not stay reddish under that situation at all. So my results, what I found is that bilirubin by itself, that's not responsible for the persistence of the red color. Also, bilirubin, if you look at it under ultraviolet light, it has a very distinct fluorescence. It's very green in fluorescence. Those type of serum edges that you see on the shroud, they're a wider type fluorescence that's not really similar to what you see with even small amounts of bilirubin when it's increased. Bilirubin is also extremely photosensitive, so that's another thing about, how might that survive, et cetera. So I personally don't really believe that bilirubin, high amounts of bilirubin can account for the reddish color in the blood. And there have also been some studies by Paulo de Lazaro where he took samples of blood from patients with high bilirubin and would hit them with pulses from a laser. So the idea that the image is caused by a energy type burst, so he would hit them with the laser, and looked at them 4 years later, and they did maintain more reddish color compared to untreated samples. But this is with colorimetric analysis, RGB type colorimetric analysis. It's not really a visual analysis with the eye. And as he points out, and I have a tremendous amount of respect for his work, as he points out, the color appearance can change depending upon the lighting and those type of situations. But a lot of what's been described for the shroud throughout the years are people that weren't using any instrumentation. They're just looking at it visually, especially outside. And the red color is something that's been noticed for quite a while. And I would also, I'd be interested in those experiments that he's done with the laser, is to see what the color of the blood is on the other side of the cloth that isn't in the direct line of fire with the lasers. Is that more like the control sample, or is that too a little bit more reddish? And similar kind of thing with the shroud. I think it would also be interesting. You don't really see that much data from the reverse side where the bloodstains soak through. Is there any kind of difference between the color of the side that's believed to be closest to the body versus that on the other side? So I would kind of, my own personal opinion, I would check no on the bilirubin box. And then it would go to the cloth idea from Ray Rogers, a member of the STURP team. He believed the cloth was treated with soapweed extract, Saponaria extract, in order to make the linen more soft and more supple. This is based on some historic writings of Pliny the Elder. So the report was that Rogers, it's not really published. No pictures were actually shown of this either, similar to Adler's bilirubin, that he could add bilirubin, it would stay red. There's no visual record of that. With Rogers, he would say that he could take blood and put it on normal linen, put it on Saponaria, soapweed-treated linen, and 30 years later, the normal blood is black and the Saponaria-treated linen is still red. Unfortunately, there's no visual record of that. It was never published anywhere. Even any photographs were included in his book or anything. So his idea is that you soak the cloth, the linen, you treat it in Saponaria soapweed in order to make it more soft and more supple. And soapweed, Saponaria, that's known to be hemolytic. So he thought that when blood would hit the cloth, it causes the blood cells to burst open, and that's why they stay red. So I tested this also. I used about 8 or 9 different preparations of saponin, Saponaria. I would also lyse red blood cells just by putting them in and out of the freezer. It's a very old school way to lyse them, but it's extremely efficient. If you just put them in and out of the freezer a few times, they will bust open. Using all the preparations of Saponaria, freeze thaw, I never saw that the reddish color was maintained, except for one particular case. It was using a saponin preparation. I just called it SAP4 because it was the 4th one that I tried. When I treated filter paper with the SAP4 solution and let the blood go overnight, when I came back the next day, it was still a bright reddish color. So I went to look at more closely what's different about SAP4 compared to SAP 1 through 9. And this was a commercial preparation. Some of them were, but this one was stored in glycerol. So when I took glycerol or glycerin, when I took that and would treat filter paper with that, I got exactly the same result. So it didn't have anything at all to do with the saponin. It was totally an effect of the glycerol. And I repeated that on the filter paper and linen for longer times. I could get the blood to stay reddish. It would look just like fresh blood for a little bit over a week, couple of weeks. And after that, it would start to turn brown, just like normal blood. So that's the only thing I've personally done that has ever caused the reddish color to be maintained. And that's something I'm very actively working on. Glycerol has a lot of break down products that are carbohydrates. So I think one or some of those could be involved in helping to keep the colors stay red.
You also talked about, I watched a presentation you gave, and you're talking about how many fruits contain citric acid and that lemon juice can prevent clotting.
Right.
And I just found that interesting. And if you could just talk about how that makes the serum halos.
Okay. So the serum halos, that was one of the things I found most interesting about the Shroud. It really caught my attention. And that was the result of, Vern Miller and Sam Pellicore did some studies where they photographed the Shroud under ultraviolet light. So when they went over there with the UV lights, they weren't really doing that to study the blood. It was a lot of the scientists on the STURP team thought the Shroud was probably some type of painting or rubbing. That was maybe first place. And second place was the Shroud was some kind of scorch from a high heat scorch from, say, a hot metal statue. Scorches from involving high heat have a very characteristic signature under ultraviolet light. So they included ultraviolet light in the studies to try to look and see if the image might be the result of a scorch. And they ruled that out, a high heat scorch. They ruled that out. But what they saw is that certain bloodstains had these serum halos or edges that fluoresced. So I thought this was really interesting because that indicates that you've had separation of blood and serum and that clotted blood has been transferred to the Shroud, a much different situation than if somebody simply added blood to the Shroud. So I was very intrigued by that. It's possible evidence here that clotted blood has been transferred to the Shroud. So I started looking through the scientific literature for serum halos, serum edges. I never ran across it. I started looking for, in the scientific literature, that serum fluoresces under ultraviolet. I never ran across it. It wasn't in there. So I did some studies where I separated blood and serum, and yes, it does fluoresce. You do get those fluorescent halos. And if you allow the blood to clot, you get it when you transfer it. If you add the blood directly, you do not. And I tried adding blood directly by a lot of different means, using droppers, using cloth to put it on, using like a piece of wood or a sharp metal point. I would never see the serum halos unless clotted blood was transferred. I let blood dry for several days. I ground it up with a mortar and pestle, added water back to it, added that to cloth, never saw a halo. I let the blood clot. I stirred it really hard, really fast. That'll break the clot up and make it liquid again. Never saw the halo. The only time I saw it was when clotted blood was transferred. So then I kind of asked, well, is there any way at all that I can get the serum halo? So that's where the lemon juice comes in. It's been known for a number of years, for quite a long time, that if you add lemon juice to blood, it will actually retard or inhibit blood clotting. And so when they do modern day collection with an anticoagulant in the tube, it's sodium citrate is what's in the tube that will prevent it from clotting. That's a related molecule is present in fruit juice, citric acid. It turns out that fruit juice is fluorescent at the same wavelength that serum is fluorescent, 365. If you put serum down on a piece of filter paper or linen and lemon juice on a piece of filter paper or linen, you really can't distinguish them based on their fluorescence. So I took a little bit of lemon juice, put it with some blood and some other fruit juices, and I found a fluorescent halo in those samples to which lemon juice had been added, similar to what you see with clotting blood. So clotted blood is not the only way that you can get a fluorescent halo. Now, would a forger have known about that? Could that have been something that they did? Possible. I think people will have to make up their own mind about that. I think if a forger had used that, you would see a halo around every single blood stain that there is on the shroud, and that's not the case. I think you also have to look at gravitational direction of a serum halo, which lemon juice additive really isn't going to give you that kind of thing. I also found that gentle heating of blood, so no lemon juice at all. If you take blood and just do a very gentle heating for 5 minutes, you'll also see a serum halo formed in that case as well. Also found that depends on the absorbency of the material. Now, we think the shroud probably would be absorbent, but originally don't really know how it might have been treated. If the material is not that absorbent, you can also get a fluorescent halo that way as well. So scientifically, I think you have to be cautious. So you can get this through clotted blood transfer, but you can also get it through additives such as the lemon juice, through heating, and also absorbencies could play a factor. So that's something I think is worth certainly sorting it out in more detail. Again, I think it's one of the most interesting aspects about the shroud bloodstains. That's something that really caught my eye initially.
There's a wet blood versus dry blood transfer theory.
Yes. So normally you would think maybe the way the blood would be transferred is just normally the blood's in a wet or semi-wet state. It transfers to the linen, as you would typically think blood is going to transfer. Some would say, well, Christ was on the cross for quite a while. There are wounds such as scourge wounds that had been there for, quite a bit. A lot of those maybe would have dried over time, etc. There is a condition known as coagulopathy, where if a person undergoes severe physiological stress, their blood remains liquid for a longer time than it typically would because a lot of the clotting factors become depleted. So there's one idea that school of thought that, well, the blood is transferred when wet, just as blood would normally be transferred. And then there's the argument, well, Christ was on the cross for a long time. They had to go get permission to get his body. That blood would have dried. So this is mainly the idea of Mark Antonacci and Robert Rucker. They believe that when radiation came out of the body, it would have taken dried blood, it heated it up into a liquid state, and then thrust that into the fibers of the shroud. And it happened that way because the blood was dry. It wasn't in a liquid state, they believed. And also that there's some areas of the shroud that weren't in contact with the cloth, so the blood could not have transferred by a normal means. So let me just say something about the contact part first. There are different models as to how the body may have been positioned when it was placed in the shroud. Some of the more modern models and artistic models, like of Blanco, that there's a hyper-realistic shroud model that's touring around. They would have the knees bent in a very severe position. Now, here in that picture, there you go. Yeah, more like the one on the bottom. Dr. John Jackson, who was the founder of the STURP team, his model, where he took data extrapolated from the shroud and also averages of volunteers in a similar position, he has it more lying more flat with maybe one leg bent up just a little bit at the knee. So it's not that kind of real severe knee bend angle that you see in a lot of the more modern models, which I think are kind of more artistic interpretation as opposed to really data-driven based on the image that's on the shroud. So one of the arguments there is, well, we would have been crucified. Those knees would have been in a crucifixion position because of rigor mortis. But what about the arms? The arms in a crucifixion are outstretched. So it said, well, rigor was broken in order to bring the arms together. Why wasn't the rigor also broken to bring the legs more relatively flat? Dr. John Jackson, there's a video on YouTube. He did a interview with the BBC. You can search on YouTube. It's called Wrapping the Shroud. So he has a kind of a styrofoam model of what he believes to be the body position, which again is relatively flat. And then he has a cloth the dimensions of the shroud, which he's put all the blood stains that are present on the shroud. And on the video, he shows how the body could have been wrapped in the cloth, and 99.9% of those blood stains line up. And I actually was able to visit Dr. Jackson a couple of years ago and had him show me in slow motion right there the same thing. He did the wrapping with that model, and it was very convincing to me. So I think the contact, no contact thing, it all depends on how you want to place the body on the shroud. If you go more toward Jackson's model, then the fact that certain areas aren't in contact is really not that much of an issue. But it doesn't really address the other point that Rucker and Antonacci make, is that, well, the blood would have been dry. How could dry blood transfer to the shroud? They claim it's through a radiation type event. So if you think about what the Gospels say about where Christ's body was placed, it was placed into a cave tomb environment. And we don't really know exactly what cave, where exactly Jesus was buried. A lot of Catholics believe it's the Church of the Holy Sepulchre. But one thing all caves have in common, and even caves here in East Tennessee, is their temperature is quite low. It's usually average around 50 degrees Fahrenheit. And the relative humidity is quite high. It's usually around 95% or so. And it's pretty much that way for caves all throughout the world. Measurements have been made in adjacent areas of the cave of the Tomb of the Holy Sepulchre, or Cave of the Holy Sepulchre, and those measurements fall within that range. So I did some experiments where I recreated those conditions: low temperature, high humidity. So what I did was I took blood, and I used pig skin for these studies because pig skin is the closest to human skin that you can get. It's what surgical students train on when they're practicing learning how to make sutures. I took blood, put it onto pig skin, and let it dry for 4 or 5 hours. I took a piece of linen or filter paper, blotted it onto the blood to verify that it was dry, there was no transfer. I then put a new piece of linen or filter paper on top of it. I sealed the edges so that no water could come in from the side. And then I put this into a humidity chamber inside of an incubator so I could adjust the temperature. So I recreated low temperature, high humidity conditions similar to what you would find in a cave tomb environment. And what I found was that overnight, after 12 hours, you had very efficient transfer of bloodstains that were originally dry. They were transferred to linen. They had very sharp edges on them, similar to what you find on the shroud. The bloodstains would also soak all the way through the material, which is also seen for a lot of the bloodstains on the shroud. And I found that not only the blood would transfer, but the serum would transfer as well. And it very faithfully reproduced what the original bloodstain was on the skin. So that's a natural mechanism for transfer of dry blood stains that does not involve radiation, but it's just a property of the environment in which it was placed. That's how you could transfer dry blood stains without having to invoke a radiation projection or something like that.
Has the shroud ever been tested for radiation?
For is it radioactive or that type of thing?
Like, have they tested for it, that any radiation?
I do not believe so. Now, there's Antonacci. One of the things he would like to do is to look at more samples to see that if radiation had hit it, it would change the isotope, the proportion of the molecules. But no, that hasn't really been done. I'm not a total expert on that, but I would probably check no on that box.
And then Ray Rogers came up with the Maillard reaction possibility, and then you came up with the composite Maillard theory. If you could just talk about that.
I will. So Ray Rogers believed that the image could be formed from a natural mechanism, and he believed it was the Maillard reaction. So for the Maillard reaction to happen, you have to have two things. You have to have a special type of carbohydrate. It's called a reducing sugar. We'll just call it a sugar. And you have to have an amine group, which is a nitrogen-bearing group. And if you put those two together, you'll form a colored product. It happens every time you brown a piece of toast. It happens every time you sizzle up a steak. That browning color that you see, that's the Maillard reaction. So this is how Rogers thought it worked. He thought that the cloth, the shroud, was treated with Saponaria, as we talked about previously, just to make it more soft and more supple. The Saponaria saponin, that's a mixture of reducing sugars. So you have the sugars present on the cloth because of the way it was treated. When a person dies, very soon after, you're going to have a release of volatiles that contain amine groups. And he thought through gaseous diffusion, those would rise up, they would react with the sugars on the cloth, and that's what would give you the image. So Rogers took linen and he treated it with saponin, with those sugars in the lab. He then exposed it to ammonia, which contains an amine group, exposed it to ammonia, and if you look at the linen, the color is very similar to what you find on the shroud. Rogers said that when he pulled out the threads and looked at them under the microscope, what he saw on those threads was identical to what he saw on the image fibers of the shroud. He took, again, another experiment. He took some sugar-treated cloth, linen. He took a hand made out of paper mache, soaked it in ammonia to let the amines soak into it. Then he put the hand onto the piece of cloth, folded the cloth over it, came back a few hours later and was looking to see if he could image the hand onto the linen. And if you look at the results, they're published in his book. I think it's just, you just see kind of a smudge there. I don't think personally, objectively, you can really tell that it was a hand there at all. There was no resolution to the image that was formed on the cloth. And that's been the major criticism of the Maillard reaction, is how can you see this kind of detail that you find on the shroud through just this sort of diffusion type mechanism. So where the composite Maillard reaction, where that comes in, it takes everything that the Rogers hypothesis had, only it now brings in amine groups which are present in proteins and amino acids on the skin. Those amine groups are present in skin proteins and skin amino acids. So if amine groups in volatile components that are diffusing upward, if those are going to react, those that are present in the skin and amino acids, when they contact the shroud at those particular points, those are also going to react. This spray tanning industry is built on that fact. So when someone gets a spray tan, what they spray them with is a solution of reducing sugar. They spray them down with sugars. It reacts with the proteins and amino acids in the skin, and that's how you get the tan. So I worked in a mouse system I did it with individual skin cells, individual hair, starting out in the paper just to show that you have the reactivity. But I wanted skin that had some kind of detail to it, some kind of resolution, to see if you really got an imprint that had resolution to it, to try to take it past something where Rogers showed with just the simple diffusion. So I would use mouse tails typically as a lot of the initial experiments I was doing. So I would take a piece of linen or filter paper. I would treat it with the sugar. Then I would take a mouse tail and place it on that. And I wouldn't leave it very long. I could just leave it for about 10 minutes. Then I would remove the mouse tail off of that. And then I would look and see what kind of image or imprint would develop on the paper. Now, if you heat it while it's in contact, you're going to see the image immediately. If you remove the mouse tail and then heat the paper, you're going to also see the image immediately. If you place the tail in contact and remove it and don't heat it at all at any time during the process, you're going to see the image. It's just going to take a few days or a week or so, a week for it to catch up, but you'll eventually see it. So on the screen, this is a mouse tail. This is a black and white negative. And I think here you can see the detail that's present, something that with the diffusion Maillard part of the reaction, Rogers could never really get any detail to come out of it. But I think here with the contact composite addition to it, You do. And I also have some figures in the paper where I took whole mice. So these are, you can buy these mice in a pet store. They're called feeder mice, and they're hairless at that stage of their development, which is why I chose them. So here's an example where we have a feeder mouse that's put down on filter paper treated with sugar, and this is the resulting imprint that you see. So if you look at the top, this is a white light photograph, and I think you can make out the eyes, the ears, and the mouth, where the legs are, etc. If you look at the black and white negative, there the details really start to pop out. And these mice have very thin, very wrinkly skin. And if you look in the black and white negative, you can pick up quite a bit of that detail of the wrinkled skin that's there. So I think these results show that you can, through a contact mechanism involving amino acids and skin proteins, it works for skin as well as hair, you can get resolution of an image. And I think if you put that together with the idea Rogers had of diffusion from vapors, which could account for regions that may not be in close contact with the shroud, I think those could produce an image. And of course, these results, they don't produce exactly everything that you see on the shroud. This is just, you're looking at just one small piece of this, a very, very tiny peek into a window to see if something like this, a mechanism like this, may be possible. But I think, personally, in my opinion, I think the results are quite encouraging. You see resolution in detail that wasn't seen before in the Maillard hypothesis. And a lot of times a contact mechanism is sort of dismissed because if you have real close contact, if you open the cloth up, you're going to have this big distortion of, say, the face area or where it was wrapped. But you don't necessarily have to put the cloth in like a vacuum seal close contact to the skin. If you just drape it over there, you're not going to really see that kind of distortion that takes place. In one experiment in the paper, I even did kind of an over-under, like the shroud would be wrapped. So I placed the tail on a piece of linen. It was long enough where I folded it back over onto the top. And when you open it up, you can see that it images both sides. You really don't need very much pressure. Just a slight bit of contact is enough for it to transfer. So I think if you have the Maillard reaction and you say, well, amine groups from volatile components can react with sugar present on the cloth, I think you also have to include the potential of amino acids and skin protein because they're going to react as well. And Rogers would always say, it's not a hypothesis that these things are going to react. If you put a sugar and an amine group together, you will get a colored product. Now, he believed that it was totally from diffusion, but I would add the skin protein and amino acid to that.
I saw this comment on YouTube. I'm going to read it to you. It says, I saw Dr. Kelly's presentation about the shroud and the possible Maillard reaction for the image in the shroud. His experiments involve skin tissue on the linen cloth, but what about the hair? Does the hair also get detailed in the image with reducing sugars and proteins?
Okay, so I've never done detailed image of hair. I've done take hair and react it with various sugars, and you'll get a colored product. But I have not done a detailed image with hair. I chose these because they were actually hairless and it was all skin. But I haven't done that with hair. I don't have any data for this, but just kind of based on how this worked, I think you would see where the hair was in contact. I think you would see a similar thing. Because if you look at isolated skin cells, if you do it on a slide and you look at hair, wherever the hair is, that's where you see the coloration. It's in those specific parts. I would say probably that would be the result, but I have not done it. It's a very good question.
What are your thoughts on the face cloth, the Sudarium?
So the connection there, is it shares a similar blood type AB. What are the chances that such a rare blood type would be shared by both artifacts? Well, I would say scientifically, it's not surprising at all that both would type as AB because blood typing with the Sudarium has the exact same inherent problems that blood typing of the Shroud has. I would like to see it verified by an independent method other than antibody or serological testing. And let's say even though they did share the same exact blood type, scientifically, to tie those two together, you really can't get around it. You would have to have DNA evidence that would link those two cloths up. I don't think even if the blood type results were solid, which I don't believe they are, I don't think that's really enough to do it. The other part of it is, well, there's blood stains that line up and are congruent, et cetera, et cetera. I've looked at those a lot to try to look at the alignment. I think some of them line up, but I think not all of them do. And even the ones that are said to line up, I personally find it a little bit questionable. I have talked to people that do this professionally, that there was actually an international meeting of bloodstain pattern analysis experts. That's what they do, is they'll look at different articles and, well, did these come from the same incident and things like that. I show them pictures of the Shroud and the Sudarium. These are their words, not mine. They said it's a hard case to make, and that scientifically they didn't really see it, and that people are going to see what they want to see. Those are their words, not mine. But I think it's difficult to make that. I think it's difficult to make that connection scientifically. Also, there's some of the matches that I've seen and some videos I've watched about, well, this is a serum part on the Shroud and this is a serum part on the Sudarium. I have seen ultraviolet pictures of the Sudarium. The serum should fluoresce. In the pictures I see, it didn't. I have some questions about, well, it's been shown that it's pulmonary fluid. I don't remember the exact numbers, but maybe it's a 1 to 10 mix of pulmonary fluid and blood. I might have that wrong. I'm not really sure, but it was said to be a mixture of blood and pulmonary fluid. So my question, well, how do you really know it's pulmonary fluid? It's based on experiments done with mixing blood and phosphate buffered saline, PBS. Well, it dilutes out this way, so it must be so many parts blood and so many parts pulmonary fluid. So I would go back to, well, how do you really know it's pulmonary fluid? Have you looked at any markers for pulmonary fluid that show that that's truly what's on the cloth? And I would think a pulmonary fluid would be pretty rich in protein, so I would expect that to be fluorescent as well, which in the UV photographs I saw of the Sudarium, that didn't really seem to be the case. Now, I haven't studied the Sudarium in detail by any means, but kind of just in general, I would think it's a pretty difficult thing at this point scientifically to link those up. I know others have different opinion, but I think if you take away the blood match, which I don't believe, again, I don't really believe the Shroud has been shown to be AB. I think if you take that away and you kind of look more closely at some of the congruence of the blood stains, scientifically, I don't know how strong the case is there, but that's my own personal opinion.
How many blood stains are on the Shroud?
Ooh, people have counted them. I could not really tell you off the top of my head. I would be guessing a number. 160. I would be totally guessing. I really don't know. But I know that people have counted they've counted every single one.
Have the tests you performed make you doubt the authenticity or the exact opposite?
Neither. So I personally, my own position is that I really don't believe enough work has been done. That's still where I stand and having done the things. I think I think that more work needs to be done before I would decide one way or the other. And scientifically, I always try to stay objective with it, doing any experiments. I try best I can, if I have an idea, to prove myself wrong, just to try to go around the problem completely 360 as best I can. As a scientist, I think it's your job to be objective. Now, personally, I have a very strong faith. I'm a Catholic Christian, but for authenticity of the Shroud, I'm right in the middle. I honestly do not believe one way or the other. I just don't think enough's been done.
Have you always been Catholic?
No, I have not. I converted as an adult. I converted when I was, my kind of tagline is I converted because my wife pulled a pistol on me. She was raised Catholic. She's a cradle Catholic. But I converted about 4 years after we were married, and since 1988. I was raised Protestant, and I just kind of Catholic faith. It was like more of a deeper expression of the faith for me personally. So that's why I converted. It was one of the best decisions I ever made.
We may have hit on this, but if Jesus' body was washed or not?
Yeah, washed. Okay, great. So that's one. So that was kind of one of the major ideas of Dr. Fred Zugby, who found that if you take a person that's deceased and they have a wound, if you wash that, it will ooze blood. So maybe that's one way that the shroud wounds, if they had dried, maybe that's one way they could have been remoistened. And that's how, after the blood had dried, after all that time, the blood got remoistened. That's how it was transferred to the cloth. So I did some experiments where I let blood dry onto skin. I used, I put blood down on the glass slides and also blood onto mouse skin for this. And I used rainwater because I thought that was more similar to tap water, since tap water has so many ionic additives. I used rainwater and just did a very gentle misting of both whole blood and isolated serum. And I didn't do any mechanical touching or actually washing of the stain. I just misted the water on top of it very gently, very low volume. I found it completely destroyed the serum halo. It's not really that surprising because serum is over 90% water. So you would expect those proteins to redissolve very efficiently and then spread out. So the halo on whole blood was completely wiped out based on glass or samples on glass and skin. And isolated serum, it was completely wiped out on samples added to glass or skin. So I think if the body had been washed, I don't think you would see those halos. If the body's in a high humidity, low temperature environment, I don't think you would see the halos either. So the idea that, well, the body was washed, I think they were doing everything in haste. I don't know if maybe there wasn't enough time for those clots to really reform before they wrapped the body in the cloth. Had they done that, based on the studies I did in low temperature, high humidity environment, there the halos don't form properly. If you take blood and place them in that environment, you will not see that halo ring form. It's arrested at an earlier stage in that blood drying development. So I think even if the body had been washed, the halo rings would not have formed properly. And according to Jewish law, I mean, the person that died that type of violent death, the body would not have been washed. So maybe Mary went rogue or the disciples went rogue and they did wash the body, but I don't believe that it was. They're based primarily, because of the Jewish law, but also scientifically, those findings. I don't think the halos would be there if they had.
Are you currently doing any tests right now?
Oh, you bet. Yeah, I sure am. Yeah, I have I'm fortunate enough to have a lab upstairs in my bonus room. So I'm doing stuff all the time. Yeah, why is the blood reddish? That's a big interest. Halos, other things. Yeah, sure am.
Any inside scoop what you're working on?
So I'm looking at some of the glycerol breakdown products that I think. Glycerol is the one thing that I got the blood to stay reddish for a couple of weeks. So I think saponin is often the one. It's kind of the one that's advertised as, well, the shroud was treated with that, and that's the source of reducing sugar. But when linen is made, it's redded by soaking it usually in pond water or lake water or some kind of body of water. I think there's a lot of carbohydrates out there in nature that could have been taken up, and you have to look at the involvement of a lot of things. So the blood redness is something I'm very actively pursuing. Also, the The serum fluorescence, that appears to change with age. So I'm kind of trying to finish some stuff up on that. One of the things that's most interesting to me about all this, it has implications for the shroud, but it also has just implications for forensics in general, which is one of the reasons I like to do this stuff, because there's a lot of stuff, general forensics you can do, and it has applications to the shroud, or there's, something about the shroud and that has applications for general forensics. So for me, it's fun to have kind of a foot in both of those.
Are you also interested in photography?
I am. So I actually have a I have a website, Tennessee Trails Photography. I take pictures mostly of animals. I do a lot of work at Zoo Knoxville, and also up in the mountains when I can get up there. I love to take pictures of the pileated woodpecker and the bears and any type of nature, but especially animals. I have taken Tennessee Trails Photography. I'll put in a plug for that. You can check it out, especially primates. I kind of take a lot of pictures of the chimpanzees, the gorillas that they have here at Zoo Knoxville, and also travel around Atlanta, like to visit around. But yes, I'm kind of photography is like a hobby. And at the school, I take a lot of pictures of events and sports events, etc. Yeah, so it's kind of a fun hobby for me to do. I was actually up in the mountains this week. Was lucky enough to see 4 woodpeckers. Sometimes you don't see them on a trip, but I was lucky enough to, within like 2 minutes of getting out and being on the trail, boom, there's like a family of 4. So the latest shot I have up there is of he's a little guy. He's like a teenager up there, but the sun is in the morning. Sun really lights up his crest. Nice. So a long answer to a short question, but yes, I like photography.
When you saw the shroud in 2015, you didn't think it could be a primitive photo or medieval photograph, or however that would make it.
Yeah, Barry Schwartz can kind of explain the rationale behind that with the light and the shadowing and things like that, how that would be off. But, thinking about how that would have been done, it's not something that would have happened instantaneous. So for a body to hang there for a few days to do that, I don't think it would be in the shape that a photograph would be. No, I don't remember seeing it. I don't really remember thinking something like that. I was probably more in the that's probably not how it was done category, even before going through. Yeah.
How can people check out your work?
So a lot of it comes out on shroud.com. Just search under my last name, Kearse, and usually it's gonna pop up there. Or the papers, you can do like a search, Kearse Shroud, and my email address is on there. You can shoot me an email. Sometimes the papers are kind of more forensics in nature, so they may not show up on shroud.com. I think the one about the washing, I'm not sure if that one's made it on there yet, but you could always get in contact with me, or most of them will pop up on there.